Hello guys, welcome to my channel and my page. And today I would like to start with the periodontics. Actually, I thought of starting with the core subject itself, but I wanted to take this topic in brief because it is covered in almost every subject, a part of it. So let's go in very brief of this blood and mononuclear phagocyte system. Fine, I'll take a very short video of this. Okay, I'm just brushing it up. Okay, so these are the contents. These are the contents which I'm going to cover in this topic. That is brief introduction to the blood, erythrocytes, leukocytes, facts about the granulocytes and the lymphocytes in detail, into the immune system, platelets, hemopoiesis, and mononuclear phagocyte system. Fine. So today I'll be dealing with a short first two topics, that is introduction to the blood and the RBC. Coming to the introduction to the blood, like if anyone asks us what is blood, we immediately tell, oh, it is a connective tissue, right? But the word which we forget here is the modified connective tissue. Why is the blood a modified connective tissue? Because there are two reasons for it. Unlike the other connective tissues, the cellular elements which are present in this connective tissue are separated by a considerable element of the connective tissue, right? This is the first point. The cellular elements present in this connective tissue are separated by considerable amount of connective tissue. And then the second difference is that some of its cells have close affinities to the cells in the other connective tissues. That is, the cells present in the blood, what are the cellular elements present in the blood, have some closer affinities to the cells which are, that is, they resemble similar to the cells that are present in the other connective tissues. That is why whenever we are asked about what is blood, we need to say it is a modified connective tissue but not a connective tissue, right? Okay, then coming to the composition of the blood, so we know that it constitutes the plasma and the cellular elements. Remember, it is cellular elements but not the cells. They are not complete cells because some of them possess organelles, some of them do not even possess the nucleus. So they are said to be cellular elements. It's more likely to say cellular elements than cells, right? And now what is all about plasma? Let us see in brief. This plasma, it constitutes about 55% of the whole blood, right? And it is the intercellular substance of this connective tissue. And other peculiar thing is that this intercellular substance is a complete liquid form, unlike the other connective tissues, right? Remember this plasma present is a liquid form, which is the intercellular substance of this connective tissue. Now, what is the composition of this plasma? That is water, colloids, crystalloids, and others. Let us see just in brief about all these four parts. Water, as we know, and then coming to colloids. Colloids mainly constitute the components like proteins. What proteins? Like prothrombin, immunoglobulin, hormones, etc. All these come under the colloids, and these colloids come under the plasma. Fine. And then crystalloids. Crystalloids mainly constitute the ions like sodium ion, calcium, potassium, chloride, phosphate, magnesium, etc. Okay, and then others. So what else are left in the plasma? These come under the others. They are glucose, amino acids, etc. So we have four components under plasma, that is water, colloids, crystalloids, and others, which constitute glucose, amino acids, etc. Fine. Now coming to the cellular elements. Under the cellular elements, we know it constitute RBC. It is not red bloods. It is more likely to say red blood corpuscle than red blood cell. Fine. WBC and platelets. Yeah. Now in this, the WBC and the platelets constitute of less than 1% of this whole blood, and the remaining, that is approximately 45%, is constituted by the RBC. Okay, so this is all about a brief composition of the blood. Fine, this is the introduction to the blood, and let us go to red blood corpuscles. Fine, so in this I'm covering a basic topics under this red blood corpuscles. Fine, I'll discuss about hemopoiesis and all in the next upcoming videos. So first let us see the dimensions. We all know, that's why we are all familiar with the diagrams of this. We see a surface view diagram, we know a cross-sectional diagram, so I have not mentioned in this video about the diagram. So let's come to the dimensions. In the dimensions, if you see from the surface view, it is a circular disc, okay, and it has a diameter of 7 micrometers. If you come to the range of this diameter of surface view, we have 6.5 to 8.5 micrometers. Fine, this is the surface view. And then if you come to the side view, in the side view you have a biconcave shape and its maximum thickness is 2 micrometers. Okay, that is about the dimensions. And coming to the structure. Now in this structure we know that the red blood corpuscles, after maturation, they become enucleated. Okay, so they do not possess the nuclei as well as the other organelles, and it is bounded by a plasma membrane. And we also know that red blood corpuscles also possess a pigment which is called hemoglobin, due to which it imparts a red color to the blood as well as to the red blood corpuscle. Okay, and the main function, we know that it has an important role in carrying oxygen from lungs to all the tissues. Fine. Now the next thing is that, okay, let's say that the blood is spread out on a film, on a glass slide. Then this RBC, which are actually red in color, will appear yellow in color. Okay, sometimes they may be red, but mostly they are more likely to appear on yellow in color. And the rims, they appear thicker than the central part. Okay, so when the blood is spread out on the film, these red blood corpuscles appear yellow or red in color with the maximum thickness at the peripheral, at the rims. And when suspended in a suitable medium, okay, when a suitable medium is selected and the blood is suspended in it, these RBCs, they pile up on one another, and this piling up over one another is said to be the Rolex formation. Fine, so that is the Rolex formation. Okay, next coming, oh fine, let us say, suppose this RBC sample is placed in isotonic, hypotonic and hypertonic solutions. Fine, So what happens when placed in isotonic solution? They maintain their normal shape, so no damage will occur to them. Suppose they are placed in a hypotonic solution. What happens when they are placed in a hypotonic solution? The cells absorb the water. And what happens when any cell or anything absorbs water? It swells. So after swelling to the maximum size, what happens? It bursts. So here it is a red blood corpuscle. So what happens when it is placed in a hypotonic solution? It undergoes hemolysis. Remember, when a red blood corpuscle is in hypotonic solution, it undergoes hemolysis. Next comes the hypertonic solution. When placed in hypertonic solution, what happens? It's obviously the opposite to the hypotonic solution. So the cell shrinks and it develops the surface irregularities, and the RBC which develops the surface irregularities is said to be the echinocyte. Okay, so you need not worry about what is this word echinocyte. Okay, echinocyte is nothing but a cell, an RBC, which is placed in a hypertonic solution. It shrinks and it develops surface irregularities. Okay, and then I'm not going deep into this hemoglobin and its synthesis, etc., that will go in deep in biochemistry. I'll just brush it up, that's it. Okay, now this shape of erythrocyte, whatever the shape of erythrocyte is maintained, it is mainly due to 4 proteins, that is spectrin, 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 ankyrin, actin, and the other proteins. Okay, the shape of erythrocytes is mainly maintained by spectrin, ankyrin, actins, and other proteins. Fine. Next, coming to the hemoglobin. We all know that hemoglobin is a pigmented one, and in this the globulin is bound to the iron containing porphyrin, which is called as the heme. Okay, heme and globulin, and this heme is an iron containing porphyrin, and to this globulin is attached or bounded, and this globulin contains different polypeptide chains, mainly 4 polypeptide chains, and depending upon the adult or fetal, the hemoglobin changes, and the different types of hemoglobins are present mainly due to this different kinds of polypeptide chains. Fine. And then apart from hemoglobin, erythrocytes also contain enzyme systems. Okay, these enzyme systems control the pH by adjusting the sodium levels within these erythrocytes. Okay, fine. So within the erythrocytes, keeping apart the hemoglobin, we have also enzyme systems. And what do these enzyme systems do? They control the pH. How? By adjusting the sodium levels within these cells. Okay, and these erythrocytes, they derive energy by anaerobic metabolism, because there is nothing to do with aerobic metabolism, because there is no a single organelle to help to go with aerobic metabolism. So it derives its energy by anaerobic metabolism of glucose and by ATP generation. This is by HMP, that is hexose monophosphation pathway. It derives its energy. Fine. So this is all about a brief, and we all know that if there is a deficiency of the hemoglobin pigment or the number of cells of RBC, then obviously it will lead to the deficiency, that is anemia. Okay, if it is more, then is erythrocytosis. Okay, fine guys, so I would like to end up this video. Just chill out. I'm giving a very small video of this, and next I'll deal with leukocytes and the immune system in the upcoming video.